Okay, I think we'll go ahead and get started. I'm your host, Andrew Morin. I'm here with about 15 other attendees in Boston in a conference with me. Nat is presenting from Berkeley, and we have about, let me check, a few dozen attendees from all everywhere around the world. So Nat is a researcher and scientist developer of software, the Phenix crystallography suite software for structure refinement. He's at Lawrence Berkeley National Laboratory, and he's going to talk to us today about the latest developments with Rosetta and Phenix and refining difficult structures. Without further ado, go ahead and take it away, Nat.
Okay, thanks. I just wanted to introduce this by saying this has been a collaboration with Frank DeMaio and David Baker at University of Washington, and it's really been a terrific experience for us because they're bringing in a very different perspective on how to deal with macromolecular structures. So the problem we're specifically trying to address is how to optimize a poor model at very low resolution. And what we need to do is simultaneously move the model into the correct electron density, refine it against reciprocal space data, but also maintain good geometry while we do that. And traditionally in the past, it's been somewhat difficult to do both at once. Usually you have something of a choice to make. And there's usually a lot of manual rebuilding involved, both to move the model and to correct geometry errors. So most of the crystallography software that that has been developed in the past has been designed to work with moderate to high resolution data. And really, 3 angstroms is generally the traditional point at which the normal methods start to break down. To deal with the lower resolution data, we need both better geometry functions and also better minimization methods. And so there's several recent examples. The deformable elastic network refinement done in CNS is a fairly well-known one, and also jelly body refinement in RefMac, which is somewhat similar things. And both of these are specifically targeted at low resolution, but also moving a model further than we could normally do that with the previous methods. So within Phenix, we have a number of different restraints for structures at poor resolution. I've listed some of these here. Basically, what these all do is they conserve the existing structure, and that's generally a good thing during refinement. But what they don't do is actually move the model to where it needs to be. And in fact, the more restraints you add, a lot of the time, the more difficult it is for the refinement to advance as far as you want it. So what we've done is combine the modeling approaches in Rosetta with our existing facilities for dealing with experimental data in Phenix Refine. And so Rosetta has a number of features that have grown up over the past 15 to 20 years, which are relatively advanced and unlike anything that we have in Phenix. And so what initially interested us was the energy function, and it's designed to be physically realistic and give you really excellent structures no matter what. I mean, it's designed to work with no data and optimize a model that in some cases is just purely a prediction. And so it includes electrostatics and very robust non-bonded restraints, rotamer restraints. But it also has some more advanced optimization methods as well. And so one of these is the repacking of protein residues to get rid of steric clashes and build rotameric side chains. And it's actually very rigorous and very picky about amino acid side chains. If they're not rotameric, the rest of the energy function doesn't work very well. So it's doing this. It's sort of a combinatorial problem, which limits how well it can scale, but it also means it's very aggressive and it's fairly more advanced than the rotamer correction we have in Phenix. And then the third element that we're really relying on is torsion angle minimization. And people have implemented torsion angle dynamics in the past, and there's CNS has one example. Phenix has an implementation as well, but that's for simulated annealing. We don't do minimization in torsion angle parameterization. And it turns out that that has certain advantages, both in terms of dealing with low resolution data, and it also seems to move things further. And then there are a couple of other features in Rosetta that are less essential, but one is real space refinement, and we do have that in Phenix as well, but it's nice to be able to do that with Rosetta's energy function. And then the other, which we're not using yet, is the ability to do fragment insertion and rebuild the structure based on a database of, I think it's 3 or 9 amino acid segments. So then within Phenix Refine, we're using all the features that deal with experimental data. So there's X-ray target functions, bulk solvent correction and scaling, B-factor refinement is called directly, and then we can generate a variety of maps as well that Rosetta uses in real space refinement. And because these programs have a somewhat similar architecture in that they're written in C++ or C at the core, it was fairly straightforward to combine them together. And so Rosetta is actually running Phenix Refine internally via Python. So I apologize again for the formatting. There are several ways to run it, but the way we recommend is using a wrapper program that hides some of the messier details of how to actually run Rosetta. And the program is intentionally meant to be run much like Phenix Refine. So you simply give it a model, PDB file, and your data file, and that's it. In the best case, you don't need any other options. There's some flags that you can specify for output, but it's designed to figure out most of the other parameters automatically. It'll extract data for you automatically like Phenix Refine. There are several other options that are useful to know. One of these is there are multiple refinement protocols. Right now, there are only 2 different protocols, and they're sort of canned recipes. And I'll show that in the next slide, what that actually means. We have one that we call low-res and one that we call high-res. And they aren't really limited in applicability to specific types of structures. The high-res refinement is less aggressive. It doesn't repack and do minimization quite as extensively as the low-resolution refinement. If you have a nearly finished structure, then there's some advantages to doing that. But eventually we want to have a large set of protocols, and you can pick the best one for each specific scenario. But there's a more powerful feature than that, which is you can actually design a script to do a custom refinement protocol. And so that's simply an XML file that's added as an additional argument, and it'll use that in preference to the built-in ones. And what RosettaScripts is it's basically an XML-based syntax for writing custom protocols for Rosetta. And this, I think, is an example of what we call the high-resolution protocol here. And it has a set of movers at the bottom, and these are all the different protocols that it's using, and then it's using each one of these as a cycle, so it'll go through n number of cycles. So in that respect, it's somewhat similar to Phenix Refine in terms of how the workflow, but it gives you much more control over exactly which steps are done and in which order. And so for the more aggressive refinement, there's actually several different types of cycle, and it switches during refinement as it goes from a poor structure to a better structure. So there's a lot of customization possible with this, more than what we can do with Phenix Refine. So one of the main limitations right now is slower to run than Phenix or most other refinement programs. A part of the reason for that is it's stochastic, and the sidechain repacking gives you different results depending on how you run it. So normally we run 5 jobs at once, and then just simply pick the best one. So these are totally independent jobs, so we can parallelize that across either a multi-processing system or a managed cluster. And so there's several different cluster technologies that we support. And basically, as many models as you're making, you can parallelize those. So the runtime, if you have sufficient resources, ends up being not too bad. If you want, the default is to use to make just 5 models, but you can have it build more models if you want. It's never going to use more processors than it needs. So if you ask for 5 models and tell it to use 20 processors, it's not actually going to run any faster. But there is the ability to sample more aggressively. There is a graphical interface now. I've just started this, so it's fairly rudimentary. It's missing a lot of the features that are in Phenix Refine, but eventually we'll add those back in. I think this is in the latest release, but it's maybe not shown by default. Anybody who's interested, I can tell you how to enable it. But eventually, this will be fully supported in very near future. So there's several practical considerations for actually running this. And so one is that I mentioned is that the repacking is stochastic. So we're always running multiple trials and picking the best. There's no real way around that limitation. We found that 5 models was usually enough to guarantee that it was going to find a good solution if it was possible. But if you simply run one, there's a chance it's just going to get stuck. And so especially for some of the more difficult structures, we would do 5 runs, and maybe 2 of them would work beautifully and 3 of them just wouldn't move at all. And it's all dependent on exactly what happens during the rotamer repacking. The other thing is right now we've really tailored it towards structures that need a lot of improvement and not giving the best possible final model. On the other hand, we spend a lot of time within Phenix trying to get the best possible final model. So we basically always, whatever Rosetta outputs is going to need some additional refinement within Phenix. I don't have the command shown here, but there's an additional flag that will instruct the program to run Phenix refine after it's finished with Rosetta, and it'll try to it'll fix some overfitting and clean up some of the bond and angle lengths. And usually it'll actually sometimes improve the other geometry aspects as well. So it's usually beneficial, but it takes more time, so it's not on by default. The bigger limitations, right now, it really works best on smaller structures. Rosetta was originally written to fold proteins ab initio, and the limit on that is still maybe 150, 200 residues. More recently, it's been used for protein design and protein docking, but it still really works best on smaller structures. We've tried running it on RNA polymerase, but it takes a while. So there's some more optimization within Rosetta that's needed to deal with really large complexes. The other problem is that Rosetta can really only handle proteins or nucleic acids at a time. You can't mix both in one structure.
We have a question here. Sorry to interrupt. Someone is asking, is it possible to use NCS to fix portions of a big model in order to stay under the 1,000 residue limit?
That's a good question. I don't know. I will ask Frank. They have features for symmetric modeling, but I'm not sure if that's implemented as constraints or restraints. And we're only just implementing constraints in Phenix now. So I think it probably would require more coding before that would actually be possible. But we should.
And it can't apply just to a portion of the model, even a smaller portion of the model using NCS?
No.
Okay.
You still have a combinatorial problem. So as far as nucleic acids are concerned, right now you have to leave them out, and that's somewhat problematic if you're doing any kind of reciprocal space refinement. There's another program based on Rosetta. I think there was actually a webinar on this already that focuses on nucleic acids, and eventually we'd like to come up with some mix of those technologies and be able to refine ribosomes and similar. And then the final big limitation is it doesn't really deal with ligands either. It has built-in support for some metals and water atoms, but any other non-standard ligands it generally has problems with. We're working on that now. I'll show an example later. I've managed to get it to sort of work, but we want it to be totally automatic eventually. And then the other thing I wanted to point out is that it's turned out to be very difficult to get both the best possible geometry and the best possible R factors at low resolution. And we're not exactly sure why this is, but a lot of the time the model with the best R factors will have significant geometry errors, and the model with the best geometry will have a slightly higher R-free. So in practice, it turns out to be a lot more difficult than I would have expected. The best possible model doesn't necessarily always have the lowest possible R-free, as far as I can tell. So what we initially focused on in our paper was improving poor molecular replacement solutions. And it's distinct from MR-Rosetta because in this case, we know it's a correct solution, and we could refine it if we had to manually, but it's quite a far distance from where it needs to be. And so we had RMSDs for the starting model were ranging from 1.5 to 6 angstroms. So there's a fairly wide radius of convergence is necessary to actually get the correct solution. So the default protocol was written to run with these test cases. And Rosetta did very well, both in terms of moving the model and in producing structures with sensible geometry. So the models from Rosetta were generally almost always the best out of all the refinement methods we tried. We tested this against a very conservative refinement strategy in Phenix that was designed to give us good geometry, but it generally doesn't move the model very far. And so these figures at the bottom of the slide are basically histograms, and the blue line is the starting model. The green line is for Den refinement, which we also in CNS, which we also compared it to. Red is Phenix, purple is Rosetta. And what this is showing here is that consistently on all of these metrics, Rosetta does very well. It's comparable to Den refinement in terms of how far it moves the model. It seems to have a slight edge in terms of R factor on average, but the big difference is really that you get really good geometry at the end. So there's fewer errors to clean up. I don't have it on this slide. We also compared it to RefMac, and RefMac was the jelly body refinement specifically, and that tends to perform comparable to the Den refinement in CNS. It moves the model quite a ways. The geometry is okay, but it's not quite as good as what we can get out of Rosetta. So this is an example from the paper. This is a fairly extreme example of the kind of conformational changes we can see coming out of this refinement method. And so here I'm comparing the starting model and the Phenix model in blue and red, and those are almost identical, basically. And then I've shown in this slide the CNS model and the published model, and you can see that you have to have a rotation of several degrees to move this domain into the right place. The CNS model has moved a fair distance. There's still some local errors, but the R-free is still 10% higher. With the Rosetta refinement, it actually snaps it almost exactly into the place it needs to be, and the R-free is very low. And actually, the R-free for the Rosetta model is lower than what was originally published. I'm comparing it to the re-refined structure in this case. This is another extreme example also from the paper, where we have a fairly large domain movement again. And it's even rigid body refinement. It was difficult to get that to move this very far. Again, Phenix gets stuck. Den moves it a little further, but it can't get the entire domain movement. And Rosetta is able to do the entire thing. And even the local details are fairly good. And as far as we can tell, the torsion angle minimization really makes a huge difference here. And that seems to be the key ingredient. Just briefly want to mention the advantage of having these more customizable workflows is if we look at the convergence of the model as a function of refinement cycle. What this is shown here is the first stage is doing purely torsion angle minimization, and it's doing a mix of real space and reciprocal space. And then the R-free kind of levels off. And then at the end, it's allowing the bonds and angles to refine freely, more like what we would do in Phenix Refine. And then you get some additional improvement in R-free. So it really is important eventually to introduce that extra flexibility in the molecular parameters. You can't just simply treat the torsion angles as rigid bodies, but it does help earlier in refinement. One other thing I wanted to mention is that what we found was that in some cases, the combination of the different refinement methods ends up working the best. So for this particular example, both Den refinement and Rosetta did very well at moving it towards the final model. Again, Rosetta had better geometry. CNS had lower RMSD to the published structure. Both of them had comparable R factors. if we took the DEN model and then refined that with Rosetta, then we actually get a significantly better model than either of those generated. The RMSD is still quite low. The geometry is excellent, and the R-free goes down significantly. So again, this is favorably comparable to the published structure. So these are very different refinement methods, and trying some combination of them may give you a very different result. So we've spent some time looking at refining models that are closer to finished. This is an example at 3 angstroms. And you can see here one of the disadvantages of the rotamer repacking is sometimes it's a little too aggressive. And so in this case, clearly the published model is approximately correct, but Rosetta really has to have very good packing for the energy function to work, and so it tends to move things out. And so there's clearly some things we can do to improve the Rosetta energy function as well, and this now gives us a framework for evaluating that directly against experimental data. So this is a more extreme example of re-refining something that's in the PDB, so not an MR solution anymore. This is one of the examples from the 2010 Nature paper on DEN refinement. And in this case, it does unusually well. I actually haven't seen any other examples where it's this big of an improvement. But basically, if we combine Rosetta and Phenix, again, we get across-the-board improvement on pretty much every metric. And it's not clear in this case what exactly is different about this structure. I suspect it's the fact that it's almost entirely helical, has some strange interplay with the Rosetta refinement function. But again, clearly there's something very different about how it's actually doing the refinement. So this is a more typical example of re-refining a published structure, and I apologize for the assaulting you with tables. But what I'm showing here is a structure that's actually fairly good, moderate resolution. We can get a significantly better model in Phenix. The one problem we can't really fix right now is rotamer outliers, and at this resolution, real space refinement in Phenix doesn't help very much. Again, with Rosetta, it basically fixes almost all of our geometry problems. And then if we do more refinement in Phenix, it actually makes it even a little bit better. We still get an R3 that's significantly lower than the published structure. We don't get one as low as what we can get with Phenix. It seems to get stuck here. So it is a better structure than a positive one by any metric. But if you're looking at R3, it's a little bit more difficult to judge. And this is another typical low-resolution example. This is a slightly more marginal structure from back when Phenix didn't deal with low resolution as well. And again, re-refining it with the current structure, with the current version of Phenix, gives you better geometry. R3 basically stays about the same in this case. But then with Rosetta and Phenix, we're able to get even better geometry. And just to mention, the Mulprobity score is basically on the same scale as resolution. So what that says is we've been able to take a 4 angstrom structure and give it the same geometry quality as a 1.7 angstrom structure. But the R factors aren't magically getting any better. If you have marginal data, there's a limit to what refinement methods can do in that sense. So finally, I just wanted to show an example with actual ligands. And this is a more moderate resolution of 3 angstroms, but the structure is somewhat under-refined. So again, pretty much any refinement method we try will give us a significantly better structure. But again, Rosetta, and in this case, I'm combining Rosetta and Phenix Refine with weight optimization. And again, we get pretty much the best model there, except slightly worse R factors. So Rosetta alone gives us lowest R factor, but the bonds and angle deviations are a little higher than we'd want, and it's a little bit overfit. But the important thing here is that I actually left the ligand in for a change. You can see there's some difference density, so it's not quite where it needs to be. But this was just run on Friday. This is not actually in the release version yet, but hopefully in the next few weeks we'll have this working a little bit more robustly. But at least it's ended up being fairly straightforward to add in support for ligands. And then, I think you've seen some of this already, but there is another method totally orthogonal to ours by Riju Das's group at Stanford for rebuilding RNA structures in Rosetta using a completely different method, only in real space. But again, they can fix geometry errors significantly, and they get better R factors. And so eventually what we want to do is combine the two approaches. So I mentioned we'll try to make the ligand parameterization as automatic as possible. We have most of the tools for that in Phenix, thanks to Nigel. There's a lot we can do to improve the map quality, and that's something Pavel has put a lot of work into. So making these maps available to Rosetta instead of the default 2FO-FC map will probably improve the performance of refinement, especially for core structures. But what we really want to do is make this as widely applicable as possible. It's not a general purpose refinement program right now. It's really it's good for special cases, but it's not a substitute for Phenix Refine or RefMac or whatever. But we would like to have as many canned protocols as possible, and we'll do large-scale testing on a PDB once we can handle ligands. And then the other thing about Rosetta is it's a very wide developer community and tackling a lot of different problems in protein modeling. And so eventually, we have access to all those same protocols from within this program. So eventually, we'd like to make more use of those. It's a lot of very advanced methods that we couldn't possibly write ourselves. And so some of these are already partially exposed, and we're going to work on incorporating those more. And I just want to close by thanking my collaborators, Frank and David. We've really been terrific to work with, and then everybody involved in Phenix, and especially at Berkeley. And with that, I'll take questions.
Okay, thanks a lot, Nat. We already have a question here from the chat box saying, is the Phenix-Rosetta Refine fully supported in all versions, including the macOS versions of Phenix?
Yes, it should work fine on macOS. Somebody reported issues, but at least on my computer, it seems to be running. It does not work on Windows. Rosetta just doesn't do Windows right now. I don't know if and when that'll be fixed. I would say if you're running Windows, you probably need a more powerful computer anyway to really run this program effectively.
Okay, and there's a question about, and maybe you covered this, there's a question about the GUI, and is there a way to pass extra Rosetta flags through the GUI during the refinement process, or are your scripts pretty much static at this point?
They're pretty much static at this point. I would say, the intent was that the XML files would be the main interface for altering the Rosetta behavior, but it's actually, it's quite easy for me to change it so it'll allow extra Rosetta flags. That's just a few lines of code that would need to change.
We have a question here. Go ahead.
So if you had an initial molecular replacement solution that you thought was an ideal candidate for this, and then a more refined one with a lot of bits cut out and a slightly lower R factor, would you suggest going back to that initial model and running this first?
It may help. I don't know if you have a lot of really poorly ordered loops. I'm not sure those are magically going to get better though. So it's probably it may be worth trying both ways, but ideally you shouldn't have to cut out too much of your model for this to work. That's the hope. I would mention if you're starting off from a homologous structure rather than the same protein in another crystal form or another conformation, then it gets a little messier. And we did try examples of those that seemed to work okay, but we had to do some additional homology modeling first. This will not fix up sequence discrepancies for you right now.
Oh.
And there's a chat question here asking about the DEN-CNS sort of combination, and they want to know, isn't DEN also incorporated into Phenix?
Yeah, there's still some issues with the performance. We haven't been able to get it working quite as well as the CNS implementation. So eventually we want to make that more fully supported, but it's been somewhat on a hiatus right now.
And there's someone asking about how well this process, the Phenix-Rosetta process, works on membrane proteins.
Well, actually, so the example, the 3FPS structure that I showed, that is a membrane protein. So, I mean, there's a large chunk of it that's actually either, well, cytoplasmic or extracellular, I forget. But the sort of helical bundle in the core that's embedded in the membrane. So that worked fine. We didn't actually have to do anything special to get that to work. In theory, you have to give Rosetta slightly different parameters if you have a membrane protein, because the implicit solvation effects don't deal well with the hydrophobic helices. But in that case, it just worked. So yes, I mean, it should work. Some adjustment may be required to really get the optimal results.
Okay, one person was interested in how to enable the GUI option. If that's easy to do, maybe you can give an explanation. If not, maybe we can do something offline for the users.
Yeah, it is easy to do. So in the Phenix preferences in the GUI, the first section is just general parameters, and there's a checkbox labeled Enable Alpha Test Programs and Features. And if you check that and restart the GUI, then additional programs will appear. The only caveat about this is we don't guarantee that your runs are going to be restorable for those programs as long as they're flagged as alpha, and we actually don't guarantee that it's not going to break. So that's why we don't show them by default. But we generally don't put them there unless they at least run for us.
Okay, and which versions of Phenix is this available in?
1.9.
Okay, well, I think that will go ahead and do it. Oh, I'm sorry, there's someone here.
Sorry, can you repeat the question?
How about the Rosetta running with the glycoprotein?
How well does it work with glycoproteins?
Oh, yeah, it doesn't right now. That counts as ligands, basically. And we don't have a way to deal with covalent attachments. So if you were going to refine a glycoprotein, you would basically have to take the glycosylation sites off and add them back later. That's a little beyond what it can handle right now. I suspect somebody somewhere probably has plans to deal with this in the future, but I don't know what the current status is.
And another question here.
What's the lowest resolution you have tested on this?
I think in the paper we looked at structures down to 4.5 angstroms. I'm trying to remember if I've looked at lower resolution. I'd have to go back and check. I don't think I've looked. I can't think of anything off the top of my head at worse than 4.5. There's not that many structures that are much lower than that. And most of them tend to be fairly huge, so they're difficult to deal with anyway.
And I think we'll take one final question from someone attending here.
Just one. I know this is mostly for academic crystallography structures. I want to know if it's also be used for like EM.
Yeah. So this program is not, but Frank is actually working on that right now. I think doing EM refinement, and we're also working on some of that separately here. So I think definitely some of the same methodologies will be applicable to EM refinement. I'm not sure what the current status of the programs is, but you should email Frank if you're interested, and he can give you details.